Okay, good morning everybody. Welcome to this presentation about the life's building blocks. Welcome to the University of Adelaide. My name's Grant Booker, I'm from the Faculty of Sciences, and I'm here to talk about opportunities in life sciences, and particularly in biomedical type areas. So welcome to the Braggs Lecture Theatre in the brand new Braggs building. It's our brand new lecture theatre in this building that's dedicated to both research and particularly teaching, and so actually it's my very first time giving a presentation in this lecture theatre, so I'm delighted to be here. Thanks for braving the weather and coming out this morning. It's hopefully going to fine up a little bit for us. So this building was named after these two gentlemen, William and Lawrence Bragg, a father and son team. They won the Nobel Prize in Physics in 1915. The father on the left was Professor of Physics here a long time ago, and his son Lawrence was born in Adelaide and educated in Adelaide University. And they went back to England and did some important work on understanding how X-rays can be used to study the structure of molecules. They really, in some ways, invented molecular biology almost 100 years ago, or around about 100 years ago. So they determined the structure of table salt and diamond and these sorts of molecules. And that has made an incredible impact on science, and particularly the science that I'm interested in, because I'm interested in the study of protein structures, and particularly in my case I'm interested in trying to design new antibiotics. So we use the structure of enzymes that are important in the life cycle of a bacterium to try and inhibit those enzymes and therefore kill that bacterium. So that sort of technology that was born 100 years ago out of Adelaide made a huge impact across the world and really underpins all of the pharmaceutical industry these days. So it's incredible what impact can come out of a couple of gentlemen working in a little old place like Adelaide so long ago. And so we're very proud to name this building after them, and it opened earlier this year, and so it's got some fantastic facilities, and I'd really encourage you to try and get on a tour to head upstairs into our brand new teaching labs. I'll talk about that later. So that's enough about me. Actually, this is not about me at all, or the University of Adelaide today, it's about you. Many of you are thinking about your futures, and you're starting to ask questions about where you want to be and what you want to do, and in particular, hopefully, starting to think about areas that you might want to study to allow you to get to that career sort of aspiration that you're starting to think about. So I want you to start thinking about the future. I want you to imagine the future. I want you to imagine a particular type of future, and that future may not be as far away as you think. And that future is sort of predicated now on our ability to sequence our own genomes. We're the first generation in history of the world ever to be able to understand their genetic makeup at the level of sequencing their entire genomes. And that is going to fundamentally change the way medicine is practiced and we interact with our healthcare system, because we're going to know so much more about ourselves than we've ever had before. And so that's the future that I want to start thinking about now. And it's happening already. So the human genome was sequenced 13 years ago or so now. It took 10 years, $3 billion to get that original sequence. Nowadays, new generation of technology, it's a few $1,000s in a few weeks to get an individual's genetic sequence. And so there's another company called Oxford Nanopore in the UK who are trying to come up with a third generation. This little gadget that's not much bigger than a USB drive. You put a drop of blood into it and you get your human genome sequence in a few hours for maybe under $1,000. And that is really going to make the difference. You suddenly can get this information and get it routinely. And I would imagine in the not too distant future that it will be utterly routine at birth to have your genome sequenced. Rather than the Guthrie test, you'll get your whole genome. And so it's a really interesting world we're going to. So we've got a situation where, in fact, this American company, 23andMe, which refers, of course, to the 23 chromosomes, you can pay them $99. Last year it was $299, I'll notice. But they will FedEx to you a little cardboard box. And you take the plastic tube out of the cardboard box and you spit in it a couple of times. And you FedEx it back to the US. And in a couple of weeks, they let you log on to a website and you can download information about hundreds, 200, 300 different genes that might influence some of your health and fitness and family tree and all sorts of other stuff. It's really quite amazing. You know, your risk for diabetes, your real risk for being a carrier for cystic fibrosis, risk for getting DVT on airplanes, this sort of stuff. And that's just the tip of the iceberg. As I say, once it's cheap enough for everybody to sequence their DNA routinely, then it will really make a big difference. And that's going on now. There are examples at this university where the group for personalised cancer medicine is interacting with the clinicians. They will take the biopsy from a cancer patient, they will sequence the cells in that tumour, they'll work out exactly which signalling pathways have gone wrong, they'll go to the cupboard, go to the catalogue, work out which drugs might influence that particular pathway, that particular mutation, and then they'll in fact go to the trouble of trying to grow those cells in the laboratory, try these medicines on the cells. If they work to kill the cell, then they can tell the clinician to use drug A instead of drug B. And that is going to really make a difference to the efficiency and efficacy of cancer therapy. And that's going on now. So it's this whole idea of you might rescue a whole lot of failed drugs that are out there already that didn't work in large populations because we didn't understand what those populations were and how they were different genetically. If we've got everybody's genetic sequence, we can say, okay, we won't test this drug in a large population, we'll test it in a specific population. And then we'll show it's safe, and then we'll show it works, and then we can use it. And so there's a whole 30 or 40 years worth of pharmaceutical research. There have been many, many failures that will be rescued because we'll understand how to use these drugs better. And again, that'll make a huge difference. And so it's really a very exciting time to be alive, I think, in this area. And the question I'd put to you, or perhaps better, the question you might put to me, is how can I get involved? And the answer is really quite simple. There's these areas, what sort of science do you need to get involved with? And so biomedical science are all the various sciences that underpin the practice of medicine and the study of medicine. I'm a biochemist, so obviously I'm a bit biased, but there's genetics, microbiology, physiology, endocrinology, a whole bunch of these ologies that get involved in there. Then there's molecular biology, and that's really crucial for this because being able to genetically manipulate, being able to sequence genomes, being able to understand these pathways that control cells, that's biochemistry, that's genetics, that's molecular biology. And then biotechnology, well, how do you take this information and make it into useful products that people can actually benefit from? Well, that's what biotechnology is. And then, of course, as I say, there's a whole range of drugs you want in the cupboard to be able to go to and then use properly. And so this idea of designing molecules, designing drugs is another important area for the future. And it won't surprise you at all to realise that we've specifically named some degrees to give you an idea about this. And so we have degrees in biomedical science, molecular biology, biotechnology, molecular and drug design. But there's also the Bachelor of Science and the Bachelor of Science Advanced. Many of you might have been in the talk before by Karen, and so you would have heard some information about those degrees. But these are degrees that we've tailored to give you a pathway. If you know where you're heading, you can use these particular degree programs and we can say, well, this is going to head you in the right direction. But it's important to realise that there are lots of opportunities and lots of flexibility here. So the Bachelor of Science itself and now the new degree, Bachelor of Science Advanced, are also excellent pathways to getting into this sort of an area of biomedical science. So I want to tell you a few stories. And so here's three interesting characters. Yin Yin is did a Bachelor of Science, Biomedical Science some years ago, and did what's called an honours degree. I want to talk about that a bit later on in chemistry. And she's now currently studying in Melbourne for her PhD. A classic example of a student who was combining different disciplines, different areas of science, both chemistry and microbiology in the case of Yin. And so she was able to combine these areas and get into her preferred drug discovery area in Melbourne. And then you've got Johan and Megan, who both did the Bachelor of Science Molecular Biology course. Megan is doing her PhD now, and she's interested actually in sex chromosomes, you know, about X and Y, males and females. That's easy in the humans. In platypus, there are 7 genes that control sex, X and Y. And so, in fact, you can tour in the Molecular Life Sciences building next door. You can tour the labs that Megan works in and see how they do that sort of genetic study. Johan, on the other hand, he's a biochemist. He's now finished his PhD. He's now Dr. Gustafsson. And he's got an interesting area because he's looking at the size of proteins at the atomic residue level. So he's a mass spectrometer. He's a proteomics expert. And so he's getting involved with this idea of personalised medicine in a slightly different way. Rather than looking at the genetic information, he's looking at the proteins that are encoded by those genes. And so he takes, say, a micrograph, a section from a tumour tissue, and there's dotted lines around some important areas, and then he uses an astonishingly expensive piece of equipment called a mass spectrometer to then map where these proteins are in this image. And he can tell the difference between individual atom levels. So if he's got a changed protein or a protein that shouldn't be there, he can detect that at precisely the position within the cell. And so he can make these sorts of images where these are not pictures and micrographs, these are quantities of proteins mapped in an XY grid. And so he can tell just how much of a particular protein is present in a particular part of a tissue. And they are often what we call biomarkers. So if you have those proteins expressed in high amounts, that's not usually a good thing. And so he can identify a particular area of it being a tumour compared to the other normal cells. And so rather than using the genetic approach, he's using a proteomics approach. And so that's an interesting area. Okay, so I've talked about personalised medicine a fair bit already, but of course that's not the only areas that careers in biomedical science and biotechnology and molecular biology can impact in. There's a whole range of them. And if you go to a few of these talks today, you'll see similar lists from different areas. And there is quite a wide range of possibilities. So really, the skills, the problem-solving skills, the critical analysis skills that you gain from an education at a university level are actually universally applicable. That's really the whole point. We tailor these courses to make sure that you get some of the specifics that you need, but in fact many of these courses could be taken in lots and lots of different directions. And so we've got lots of students are aiming to get into medicine, and they come through and do graduate medicine, so they do the underpinning science with us and then go to a graduate medical school elsewhere. We've got defence industry, forensic science, food technology, teaching, the list goes on. And in fact, this is not an exhausting list. There's lots of people out in the marquees that you can talk to and ask them about their careers and what other options are available to you. So I thought I'd also just give you a bit of a feeling for the course structure. So maybe you've decided that this is an interesting area and you might want to come along and see how you can get involved. And so what would you do? Well, you'd enrol in one of these courses, enrol in one of these degree, which we also call programs. And so how many courses are involved? Well, most of them are 3 years of study. And in first year, we have 4 courses in semester 1, so 4 courses in the first half, 4 courses in the second half in blue. I don't think that's me. We've got, so it's a total of 8 for the year. We've got another total of 8 for second year, second level. And then you'll notice that we go to fewer, larger courses in the later years, in third year. And that's so that we can start with breadth, we can start with teaching you lots of stuff about lots of different areas, and as you get closer to where you want to be at the end, we start to specialise, and so you can go deeper into the areas that you want to study. And so we tend to have fewer subjects, fewer courses, but go into them in much, much more detail. And then after 3 years of study, and all's gone well, you can graduate with a Bachelor of, say, molecular biology. We then find about half of our students, about 50%, go on and do a year of further study that we call an honours year. And I know it seems a long, long way away, But really, I'd start encouraging you to think about even doing that 4th year from the start. Because I find that most of my students learn and gain more out of that final 4th year than all of the other 3 years put together. It's a research year, research intensive year. You spend the entire year in a laboratory with professional scientists on the project. You then write a thesis on that project. There is some other coursework and so forth depending on the particular honours degree that you do, but most of the time is spent doing real research with real research scientists. And so it's a great way of learning about problem solving, critical analysis, communication, these sorts of skills, and you can take those anywhere. So I know it's a long way off, and it's a big commitment, 4 years, but just have it in the back of your mind that maybe after 3 you might sign up for the 4th. There's a whole lot of stuff you can do, masters, PhD, let's not even worry about that today. But I really strongly encourage to do that. So what sort of courses we do? Well, one of the other hats that I wear is as Level 1 Director in Sciences for Biology. So I oversee all of the biology courses in the sciences at Level 1. So we start off with a combined class called Molecules, Genes and Cells. And not surprisingly, it's about molecules, genes and cells. It's about 1,000 students in the class. So it's a new world in university. You have to get used to big theatres like this and being sort of one of many, many, many heads in an audience. And so we then, in second semester, split into 2 alternative courses, Biology of Organisms, which as the name suggests, it's about plants and animals and how they interact with their environment and their structure versus their function at the whole sort of organism level. And then the alternative is Human Perspectives, which obviously is biology about humans. And so that's where a lot of the biomedical science areas kick in. And so after that, grounding in biology, it all explodes in second level. There are huge numbers of biology courses. You start to specialise a bit more. Biochemistry, biomedical science, biotechnology, genetics, microvirology, physiology, you name it. There's a whole bunch of them. I've probably even forgotten some. And so the opportunities really balloon out into lots of directions, and so we can almost satisfy any of your interests in the biological sciences. One of the comments that I would make, though, is that you're in the situation, many of you now, about thinking about subject choices. So some of you might be in Year 11, some might be in Year 12. But if you're in Year 10 and you're thinking about your Year 11 subject choices, I'd really strongly encourage you to keep your options open and broad. And what does that really mean? Well, it means chemistry, maths, particularly, maybe some physics, maybe some biology. My colleagues will kick me for it, but I'd actually much rather you, if you had a choice, if you had to decide in Year 11 or 12, particularly perhaps 12, do I keep chemistry or do I keep biology? Maybe you can't do both. Please do the chemistry. Please do the chemistry. We think we can teach you the biology much more easily if you've got the chemistry background. All the stuff I've talked about this morning really relies heavily on this molecular knowledge of biology. And the more and more in the future, it's going to be quantitative. And so understanding how to do some maths is going to be very valuable. It's going to be less descriptive and more quantitative. So please try and keep that maths. I know it's not always fun, I know it's not always easy, but please try and keep that maths as long as you can and keep that chemistry as long as you can. The physics is also really great. The biology, we think we can do that once you get here. So lots to think about, lots to see and do. Here's Sam and Felicity in our brand new labs up on the 4th floor of the Braggs building above us somewhere, quite a few flights. And they're probably playing Angry Birds, but I don't know. So please grab on to one of the tours and head up to the 4th floor and have a look there. There's lots of things to do and see. We've got some activities for you. We've got the marquee tour next door. There's a whole bunch of science marquees and lots of people desperate to tell you about what they do and how they can help you get to where you want to go, because after all, that's what Open Day is about, giving you the information that you need to get to where you want to go. I think I jumped a slide. There's an open night on the 11th of December, and so come along and see a little bit about university life, I would argue, and I've been around a while now, I would argue that it's probably the best time to be coming to university in the last 20 or 30 years. Why do I say that? Well, this building's one of them. It's a fantastic facility for learning science, for learning biology and physics. We toured the country when we designed the labs, and we made sure that we took the best of what everybody else has got. And so, having opened only just this year, I can say pretty confidently that we have the best teaching laboratories in the country right now. So that's why I encourage you to go and see them, because they really are fantastic. But also, if you haven't been to Hub Central, that is the best student space in the country too. And again, I've seen a few around the place. And there's some good ones, similar style to ours, not as big, not as fancy. Just at the moment, that's the best student-oriented space in any university in the country. It's fantastic. In Cargill Waterley, next door is a magnificent building. So we've invested an awful lot in our infrastructure lately, and that is going to make your study here much more enjoyable. So I'd really encourage you to have a look at those facilities and ask these people what they do, because they'd love to tell you. So visit the marquees. A colleague of mine is going to be taking tours to the 3D suite over in the Santos Petroleum Engineering building, and you can look at some of these interesting molecules with glasses on and see them in vivid 3 dimensions. It's a lot of fun. And so shortly outside there'll be a tour. I'm sure there'll be a flag waving somewhere for you to see it. So I'd encourage you to get onto those tours. And I hope we've just given you something to think about for the rest of the day and for the rest of the week. And I thank you for listening.